Hello and welcome everyone. Thank you for taking the time out of your busy schedule to attend this webinar today. I'm Keshav Vasantavada, your host and presenter, and I'll be discussing the role and importance of recombinant antibody production within the therapeutic antibody discovery context. Quickly about me, I'm the Senior Marketing Specialist for Protein Services at GenScript. Prior to joining GenScript, I worked for many years in a big pharma in the research facility, doing molecular biology and protein biochemistry work, including expression and purification of a variety of disease targets to support the therapeutic areas on site. From my own experience and the experience of my colleagues and clients, I learned that recombinant expression is a bottleneck in many labs. And so in line with that experience, today's webinar will discuss some aspects around the recombinant expression of antibodies and the technologies and capabilities that GenScript offers that would allow you to break the bottleneck of recombinant antibody expression. In doing so, I'll also highlight some services that will potentially match your needs and your downstream applications. We'll also discuss the role of high throughput methods for recombinant antibody production that could potentially accelerate your antibody drug screening process. A few housekeeping items before we begin. I assume that you can all see my screen right now. As we move along, if there are any questions, please type them in the chat box. I'll answer them at the end of the session. And for those questions that I'm unable to answer for lack of time or ability, a representative from GenScript will follow up, discuss personally. And by the way, this webinar is being recorded and it will be available from our website for viewing later. So with that, let's move on. So here's the outline of my presentation. I'll do a quick introduction about GenScript and our track record before diving into the subject of recombinant antibody production. We'll then take a look at GenScript's technologies, services, and our capabilities in recombinant antibody production. That will be of value to therapeutic antibody researchers. We'll look at a few case studies that will illustrate the strategies that our production team utilizes in enhancing recombinant antibody expression. And we'll also review an example of the application of high throughput antibody production approaches that has enabled our clients to speed up their antibody optimization and screening campaign. So quick intro about GenScript. We're headquartered in Piscataway, New Jersey. GenScript is the world's leading biology research service company. We are internationally recognized as a leading contract research organization specializing in biological research and early phase drug discovery services. We provide services and products to scientists in 86 countries around the world. We have built the best-in-class capacity and capability for biological research services encompassing gene synthesis, molecular biology, peptide synthesis, custom antibodies, protein expression, antibody and protein engineering, and in vitro and in vivo pharmacology, all with the goal to make research easy. More than 13,000 peer-reviewed journal articles have cited GenScript's services and products, making GenScript the most frequently cited biology CRO in the world. GenScript offers expression services in four different hosts: bacteria, insect, mammalian cells, and yeast. Today's webinar will focus entirely on recombinant antibody production in mammalian systems. But while you're on this slide, I wanted to let you know that we do offer antibody fragment production services as well. We produce the variable heavy domains and the single chain variable fragments in E. coli using our FragPower service, and we can produce the fabs in insect cells as well. And these names, beside each expression system, they are the various packages, various services that we offer relevant to that particular expression system. Track record, and to quickly give you an overview of our production team's track, they have delivered more than 6,000 batches of high quality proteins and more than 1,500 batches of purified recombinant antibodies till date. Our customer base is over 40,000 in more than 80 countries around the world. Our aggregate success rate for all projects is more than 92%. And when it comes to recombinant protein, we have successfully produced a variety of target types, as you can see in this chart below: kinases, cytokines, GPCR ion channels, transferases. You name it, our production team has had the experience to produce it successfully. And here's a snapshot of GenScript's antibody drug discovery services. As you can see, we offer end-to-end solutions for your therapeutic antibody projects. GenScript has established a very comprehensive antibody drug discovery platform, which includes antigen production, hybridoma development, epitope mapping, antibody sequencing, antibody humanization, affinity maturation, related functional assays, tumor model development, and recombinant antibody production that can meet all your antibody drug discovery needs. In the remainder of this webinar, I'll focus on the key production aspects of recombinant antibodies as applicable in a therapeutic antibody context. Okay. So moving on to the topic of recombinant antibody production. So why recombinant antibodies? As you all know, monoclonal antibodies are ubiquitous in research and medicine. The traditional monoclonal antibody technology, the hybridoma technology, is nearly 4 decades old, and it's a very mature technology. Plus, it's very robust and cost-effective. But there are issues with its use. Some of the issues include ethical concerns, non-specificity issues, and the instability of hybridomas themselves is a problem because they can cause the loss of antibody genes. Now, some of the more significant issues include potential immunogenicity, as in the case of HAMA response. So as you know, a vast majority of patients that are treated with murine antibodies produce the HAMA response, or the human anti-mouse antibody response. Antibodies produced by the HAMA response can neutralize or result in the clearance of therapeutic antibody in immune complexes, or they can even sensitize the patient to allergic reactions upon re-administration, and that can compromise the safety of the antibody therapeutic. Now, chimeric antibodies have partially solved this problem, but nearly half of the patients that are treated with chimeric antibodies can develop the HACA response, or the human anti-chimeric antibody response. Now, so humanization of antibody and subsequent recombinant production can offer a solution to these problems. Another significant problem with conventional antibody is the lack of reproducibility, as was highlighted in 2 different Nature articles published last year. In those articles, the authors discussed the reproducibility crisis in biomedical sciences and how it was caused because of antibodies that fail to do what they're supposed to do. And the authors estimate that approximately $350 million each year is wasted on antibodies that are not reproducible. And so these findings, combined with other observations in the research community, have caused a massive shift towards the use of recombinant antibodies. So much so that scientists at leading organizations, including over 100 co-signatories, have proposed a shift entirely towards the use of recombinant antibodies. So we know that there are problems with existing technologies, but then what are the advantages of recombinant antibodies? And on this topic, I just want to let you know that we are talking here about the recombinant antibodies in the context of bioproduction and not lead generation. And the reason I'm making this distinction clear is that when you think of recombinant antibody technology in drug discovery, one would immediately think about the display technologies, just phage or yeast display. And while that's a purely recombinant technology, its purpose is different. Recombinant antibody display technologies, as you know, are for obtaining high affinity lead candidates that enable the development of antibody therapeutics. In the current context, we are discussing recombinant antibodies in the context of bioproduction, which is essential for screening, optimization, and characterization of such leads. Okay. So now, having made the distinction clear, let's look at the benefits of recombinant antibodies. So as you know, recombinant antibody technology is entirely non-animal, so there are no ethical concerns. You have more control over the process. It's really rapid. Production time is in weeks as opposed to in months. It's amenable to high throughput processing. It can be constructed against virtually any antigen. Immunogenicity is not an issue. And recombinant antibodies can be produced in several formats. So you have Fabs, single chain variable fragments, diabodies, etc. Excuse me. And they're amenable to fusion with drugs and toxins. And since they're sequence defined, they're more reliable and more reproducible, and they're more conducive to optimization and engineering. Excuse me. And while there are reasons to utilize the recombinant antibody technology, it is important to note that recombinant antibody technology for bioproduction is not intended to replace conventional technologies. In most cases, recombinant antibody production technologies supplement the existing technologies and existing platforms. So let's look at the entry points into recombinant antibody production. So as you can see from this slide, a researcher can enter the recombinant antibody workflow from any platform. So for example, hybridomas. Coming from hybridomas, the typical workflow would involve identifying the sequence of your antibody by sequencing the hybridoma, and it's a first step towards antibody engineering and functional optimization. And it's also critical for downstream patent application. And while here, I just want to let you know that GenScript advanced technology offers fast and professional sequencing services for your hybridoma clones. And to utilize this service, you simply submit your hybridoma samples to GenScript, and we can sequence it for you. And if required, we can do the gene synthesis and recombinant antibody expression as well downstream. If you're currently using phage display, then more than likely you would have to convert your lead antibody fragments into a full-length antibody format, which would be more suitable for in vitro and in vivo applications. And this would be a good place to switch to recombinant antibody production techniques so that you can utilize the power of gene synthesis to create full-length antibodies from your fragment sequences, and then funnel that for recombinant expression. You can bring in leads from transgenic mice into a recombinant antibody production workflow, and there are other ways in which you can utilize the recombinant antibody technology, and I'll show you that in a case study a few slides later. So let's look at GenScript's capabilities and services that can match your needs and your applications. So now that we understand the importance of recombinant antibody production in supporting antibody discovery platforms, let's see the various options for producing recombinant antibodies. Now GenScript offers transient expression, stable cell pool, and stable clonal cell line technologies. Each has its distinguishing features. Transient expression is good if you have relatively smaller requirements for antibody, and if you need it really fast. As little as microgram quantities and as much as gram level recombinant antibodies per liter are possible with transient expression. GenScript can provide transient expression of antibodies in as little as 6 weeks at a very competitive price. Our high throughput antibody production packages start under $500 per antibody, and I'll present the details of that service a few slides ahead in the webinar. Stable cell pool, it's sort of a midpoint between transient expression and stable clonal cell line. Now the yields from stable pools are typically 4 to 5 times higher than transient expression, and so it's a great alternative for antibody reagent generation. Another advantage is that stably transfected cell pools can be frozen down, and they can provide working stock for further production, if you ever need them. So you don't have to retransfect cells again. And the stable cell pool is a good option because if you're on track to develop a stable cell line, but want to have some reagent really fast, then stable cell pool comes handy. In our hands, expression level from stable cell pool, can be as low as 300 milligrams and as high as gram level. And turnaround can be as fast as 8 weeks. And I'll present some more information about a stable cell pool service ahead. Stable clonal cell line. I forgot to cover that. As you know, stable transfection also begins transiently, but through a process of careful selection and amplification, stable clones are generated. And in stably transfected cells, the foreign gene becomes part of the host genome, and so it's replicated along with the host, resulting in a stably transfected cell line. For large-scale production of therapeutic antibodies, stable gene expression is a preferred choice, as it allows for greater process consistency and control of the final product quality. And I'll also highlight GenScript's latest stable cell line offering in a slide ahead. Transient expression. So large-scale transient transfection is a way to speed up the production of milligram to gram quantities of recombinant antibodies and proteins in mammalian cells. And as you'll know, CHO cells are usually the preferred option for stable expression, but HEC293 is a popular choice for transient transfection. But GenScript has in-licensed the technology, transient expression technology, from the National Research Council Canada, which allows us to get comparable yields both from CHO and HEC293 cells. And as you can see in this graph, the blue line is the antibody titer in 293 cells, 7 days post-transfection. It's close and comparable to the antibody titer from CHO cells, the orange line, using an optimized process. So the NRC technologies come with the processes, the cell line, and a special family of vectors called the PTT vectors. And this system combined enhances expression levels 3 to 4-fold when compared to conventional systems. And as I said, GenScript has licensed this technology from the NRC Canada, and these cell lines both of them can be used for rapid and high yield production of both proteins and recombinant antibodies really rapidly and in serum-free media. Stable cell pool. So as I described earlier, stable cell pool is like the midpoint between transient expression and stable clonal cell line. Excuse me. Yields from stable cell pools are typically 4 to 5 times higher than transient expression. So it's a great alternative for reagent antibody generation. And GenScript we use our glutamine synthetase expression technology for producing stable cell pools. In the various projects that we completed recently, as you can see on the upper graph, our titers range between 300 to 400 milligrams per liter all the way to gram and up. The timeline for stable cell pool screening is shown below in this table. So it involves preparing cells for transfection, which takes about a week, and then transfection and cell pool screening takes about 4 weeks. The cell pool evaluation is a couple of weeks, and then prepare cells for delivery. All in all, about 8 weeks is the timeline. So really, really rapid. And then take a look at the GenScript stable cell line technology. So we recently launched a glutamine synthetase-based expression cell line for stable expression of recombinant antibodies and proteins. Now, as you'll know, there are two predominant expression systems in the market for stable expression of antibodies. One is based on DHFR selection, and the other is based on GS, or glutamine synthetase selection. So this is a GS-based selection system, which offers high productivities without amplification. It's really rapid. There is no need for rounds of gene amplification. It's a very scalable process. The cell line is CHO-derived. It's adapted to suspension in animal-free chemically defined media, and it's selected for superior growth and expression. And we have a cell line development process, protocolly optimized process. We can achieve product concentrations of more than 5 grams per liter with a high probability of monoclonality. And if you look at the chart below, while the maximum expression levels are, really depend on the product, we have created cell lines that can produce more than 5 grams per liter recombinant antibody with a production range in the 50 to 100 PCD, picograms per cell per day range. And you can see in this chart, we can go from pool to round 1 fed batch. We can already achieve more than gram per liter expression, and taking it into round 2 fed batch, we can go beyond 5 grams per liter. So in this technology, we use a robust expression vector and selection by glutamine synthetase metabolism, to provide these high-yielding cell lines. And as I said earlier, this process is really scalable. So yeah, and this is really important because when you go, for example, in a manufacturing run, increasing volumetric productivity is really important to cut down the cost, and that's when a cell line like this comes in really handy. So that's about our latest cell line. And then this is a general schematic of our recombinant antibody production workflow. If you're wondering, okay, how, if I need to use your service, what do I do? It's very simple. The process begins with providing your sequence. GenScript takes over from there. We do the codon optimization, gene synthesis, construct generation, transfection, expression purification, and we deliver the purified material to you. Really simple and elegant workflow. And here I want to show that GenScript offers recombinant production services that can integrate well into your pipeline, going all the way from discovery to preclinical development to GLP workup and GMP workup. And in the discovery phase, we have protein and antibody services that are applicable to the various stages: target validation, screening, lead generation, optimization, characterization, and leading all the way to the selection of your lead antibody. And I've listed a few examples here. If you look at the numbers on the top and the table at the bottom, you can get a flavor for the kind of services we offer. For example, if you look at 1 and 2 that are suitable in the discovery phase, they are the high throughput expression and purification of recombinant proteins and antibodies. We do them in 1 to 5 ml volume, 96-well plate format. Purify the antibodies with protein A, resin, and give you purified material back. We do lab-scale transient expression in shake flasks, which is suitable for both discovery and preclinical work phases. And if you look at 3, 4, and 5, it's mid-scale expression, both transient and stable pool production. We can do large-scale weigh bags or bioreactors. And then we also have very extensive characterization services with LC-MS capability at the core. And as you know, it's really critical for verification and the release of bioproducts, and it's more important in the latter stages of R&D. So we can integrate our services into your pipeline quite easily. And this is another service guide. This information is available on our website, so you don't have to go through all the details right now, but basically what this is I list the purpose here of your antibody expression in this first column. That could be screening, candidate validation, in vitro efficacy, in vivo efficacy, and safety testing. And then we have a bunch of services that are suitable to each stage along your workflow. And I list the production type and the volume, the antibody quantity that we can get from each of these services, the timeline, and the guiding price per unit, and the QC standards. So you can see that, given our expertise in antibody production techniques, we provide a comprehensive suite of services that can deliver from microgram to gram quantity, multiple gram quantities of pure antibody for each stage of your drug discovery campaign. And if you look at the flow here, the summary is as you go down the pipeline, the QC standards go up and the guiding price per antibody unit comes down, given the economy of scale. So as I said, this information is available on the website, so please feel free to view this in detail later. And I also want to tell you about another guaranteed antibody production service that we offer. It's called MamPower. And this is meeting the increasing demands for high quality recombinant antibody production for many of our clients. Using this package, the antibody amount and purity are guaranteed, and endotoxin control is also performed. So customer simply needs to provide the antibody sequence and we can do the rest. Final deliverables include purified antibody at the listed amount and the purity, and the light chain and heavy chain synthetic genes and PUC57 vector, the optimized gene sequence report and QC data are also delivered. So this package offers a rapid turnaround going from sequence to purified antibody in as little as 5 weeks, and the starting price is under $3,000. The high throughput gene to antibody workflow. So this is something that we launched relatively recently. It's our high throughput antibody purification service for the rapid production of recombinant antibodies for your antibody drug screening and optimization campaigns. So basically the service begins with customer supplied sequence, and the end deliverables are purified recombinant antibodies, QC data, and a comprehensive expression report. There is no setup fees for the service, and gene synthesis is included in the price of this service. So we have two different scales, a 50 microgram scale and a 1 milligram scale, and the starting price is $4.99 per antibody, and it includes the price for gene synthesis. So basically, as the workflow demonstrates, there's two different ways you can utilize the service. You can just provide your antibody sequence. If you do that, we just do the sequence evaluation, codon optimization, high throughput gene synthesis, which is the cloning, plasmid prep, transient transfection, expression screening, one-step affinity purification, QC, and we give you the purified antibody. But if you already have your DNA, synthetic DNA, you just provide the DNA and your antibody sequence evaluation, and we do the rest for you. And this new service, high throughput service, is designed to help researchers scale quickly from promising leads to purified recombinant antibodies in a microscale format, to accelerate their preliminary drug screening process. And some of the applications that are served using this service include epitope binning, ligand competition binding studies, affinity measurements, primary and secondary assays, high throughput by physical characterization to assess developability, and much more. And another good thing about this service is that any promising leads from your high throughput campaign can be seamlessly integrated into our workflow for scale-up transient recombinant antibody production or stable cell pool or cell line production. If you need milligram or gram level expression downstream to support your in vitro and in vivo applications, this can be a good starting point. So with that, let's take a look at some of the case studies. So this is a case study where we had low expression to begin with. And this case study illustrates how efficiently we could increase the expression levels. So basically this order was for the 50 milligram guaranteed antibody production, the MamPower that I talked about a few minutes ago. So when initial expression evaluation was conducted, we found that the expression level was really low, and we achieved only 2.5 mg per liter. expression level. So to obtain 50 milligram antibodies, you would need a 20-liter culture volume. So the increased expression level, our production team designed the antibody sequence, performed codon optimization, and used a special signal peptide and optimized the transfection purification process. And at the end, we achieved 25 milligrams per liter expression level. So we could obtain 50 milligrams from just 2 liters of culture volume. And these are the gel images, control and purified antibody, both reducing and non-reducing lanes. And if you look at the second case study, this is no expression at all when we first started the project. So if you look at the left lane, it's a Western blot image. This is a standard IgG case study. Lanes 1 through 6 are reducing lanes, and the 7 through 12 are non-reducing lanes in 293 CHO and 293 CHO. But really, what's important is no expression when we began, but after sequence revision and subsequent optimization, if you look at the right side panel, we could obtain expression levels as high as 15 milligrams per liter. If you look at the left 6 lanes are reducing, the right 6 lanes are non-reducing. So we do see some intermediates and some higher order aggregates, but we were able to purify the required antibody 15 mg per liter expression. So another successful case study. And let's look at another case study where our client has utilized our high throughput recombinant antibody production technology to speed up their antibody screening process. So to begin with, the client has access to proprietary technology for identifying and isolating antigen-specific B cells. So using a group of 4 patients, there are 4 healthy immunocompetent patients who had contracted and cleared the HIV infection, they identified the B cells specific for the desired epitope, isolated and sequenced the unique antibody sequences, cloned, purified, and confirmed the activity of antibodies, and then engaged GenScript to continue preclinical work on a panel of positive antibodies. And so this client is focused on the biology and developability of their lead antibodies to ensure efficacy while minimizing any downstream risks. So that's the context. And let's take a look at what was done. I know this is a very complex slide, so let me walk you through it. And I can tell you how our services allowed the client and supported the client in their screening and candidate validation steps. So this is our client. They identified 4 different antibodies from patients. They're anti-HIV neutralizing antibodies. So 1, 2, 3, 4. So they have their own proprietary algorithm using which they can in silico optimize the sequences for further development. So that they do that and they supply the sequences to us, and then we do the gene synthesis, codon optimization gene synthesis, and then we express those antibodies, purify the antibodies, and give it to them, and they perform a bunch of high throughput biophysical characterization on those antibodies, and they identify about 10 to 12 that are active and they have the desirable biophysical properties, and then they go on to do further in silico work, optimization, give us the sequences, we do the high throughput antibody production, give them the antibodies, and then this cycle repeats a few times, and from this exercise emerge a panel of about 3 to 10 antibodies, and at that point the client would need a few hundred milligrams for a variety of biophysical tests, viscosity screening, colloidal stability, solubility stability, screening assay filtrability, and they need more antibody, few hundred milligrams for some animal studies. So our, the GenScript logo that I show here are the various places where our services come into picture. So the high throughput antibody production service, we have our MamPower guaranteed antibody and our customized antibody production services that come into play here. So after this work is complete, then the client would need a few hundred milligrams for their in vitro neutralization assays, and a few grams for in vivo efficacy as is in their safety tests. And so we have our MamPower guaranteed antibody, large-scale antibody, and our custom antibody services, and our cell pool and cell line services that come into picture here to meet all our clients' needs and their applications. So with that, let's go to the summary and conclusion. As I said at the beginning of the webinar, our production team scientists have successfully purified and delivered over 1,500 batches of recombinant antibodies at a variety of scales. And so scientists have accumulated experience in producing a variety of antibody formats, including IgG, IgA, IgM, the bispecific IgG, single-chain variable fragments, bispecific scFps, and Fc fusion proteins. Our scientists can perform a variety of optimization experiments and leverage their years of collective experience to achieve high yields, excuse me, and deliver high-quality antibodies to the clients. Unfortunately, due to time constraints, I cannot present case studies of all these antibody formats. And what I presented today is just a small sample of our overall capabilities. But I invite you to visit our web pages to learn more. In our technical notes section on our web pages, we have added more information about some of these antibody formats along with some production data. So our quotations and ordering workflow. So if you're considering, reaching out to us for our services, you might be wondering, how do I do it? So our workflow is really simple. Almost all our web pages have a quotations and ordering workflow at the bottom of the web page, and we have a quote button on the upper and lower right-hand side. All you have to do is click that get a quote button, provide your information, and submit. And our technical account managers will contact you to finalize the quote. You just review the order details and the price, provide your credit card or PO information, and your order is placed. And then after the order is placed, our project managers will be in touch with you to provide timely updates and communicate with you. And if you have any questions, you can certainly shoot us an email, and there's other ways you can reach out to us, and we're always eager to help. So another thing I just want to close, before closing, just mention why choose GenScript. So if you were to ask me, Keshav, why should I choose GenScript's antibody services, 5 top things that went to my mind would be quality, speed, price, expertise, and support. So if you look at quality, it's not just the quality of deliverables, it's the quality, it's the complete package, the quality of project management, communication, along with deliverables. Speed is crucial these days, and working with GenScript will give you the speed advantage. And not just because our turnaround time is really fast, but also because we are the largest synthetic gene supplier in the world. And so we are truly a one-stop shop, going all the way from sequence to purified antibodies. Now this is an advantage that many of our competitors do not have. And this is really important because working with a single vendor will greatly minimize the overall timeline and your downstream risks. And in case a problem arises ever with any of your projects, troubleshooting is much easier when you're using a single vendor like GenScript. Price. So, as you saw in some of the earlier slides, we are really price competitive, and most of our prices include the cost of gene synthesis. For example, starting from $4.99 for antibody production, high throughput antibody production, starting from $2,999 for guaranteed antibody production. These are really competitive prices. Number 4, expertise. As I showed earlier, our production team has literally worked on thousands of proteins and antibodies. So that's a lot of accumulated experience and expertise over the years that we can lend to your projects. And lastly, support. Technical support team consists of PhD level trained lab scientists. They're just a click away in case you have any questions. And our marketing, sales, production, project management teams. They work closely with the technical support staff to make sure that you have the most updated and most accurate information whenever you need it. And I can also tell you my own customer experience. Before I was a GenScript employee, I used to be the customer in the industry. And when I ordered from GenScript, things would always arrive on time, and they were really high quality deliverables. But I never really put thought into what went on behind the screen. And now, from a GenScript point of view, I can lend you my customer experience and GenScript context as well. When you place an order, there are many things that go on the backstage. That it's like a really fine clock coming together to make sure that you get your order in the requested time at really high quality. So there's a lot of integrity that goes into each order. So I can assure you that your projects are in safe hands. And and so just before we move on to the Q&A, just another note is, assuming your lab is involved in antibody drug discovery work, you might be interested to know that GenScript recently launched a concise and colorful handbook that presents topics around recombinant antibodies and antibody drug discovery in bite-sized nuggets of useful content for easy reference. Some of the topics include antibody structure, antibody formats, fragments, the antibody drug discovery process, production topics, and more. So you can download the electronic format of this book from the website listed here at the bottom. And this is a slide that shows our upcoming webinars. So you can sign up if you're interested. So we have a couple of they range in topics from gene topics, CRISPR, recombinant protein production, and insect expression. We have a couple of antibody related topics, in April and May. So please do sign up. And as I mentioned earlier, at the beginning of the presentation, this webinar will be archived on our website, which is shown here. The URL is shown here. So please do visit our webinar page to take advantage of these resources. And if you have any questions or feedback for me, you can reach out to me directly via email. My email is listed at the bottom of the slide here. And also, it would be great if you could take a moment to complete a poll at the end of this webinar to tell us how you like this presentation. Your candid feedback will help us improve our services. So with that, I guess we can move on to the Q&A session. So if you can just bear with me, give me a minute here. I'll just go through the list of questions and do my best to answer them. Just give me a minute. I'm scrolling through the list. So what is the pricing structure for your MamPower packages and what's included? It's a good question. It's our MamPower guaranteed antibody package starts under $3,000, $2,999 to be exact. It's for the 5 milligram antibody production, and there are various pricing tiers. We also have 10 milligram package. It costs about $3,500, and we have 50 milligram package, which is about $7,500, and we have 100 milligrams and 500 milligrams, and so on. So you can get a quote from our webpage, and basically you just need to provide the antibody sequence, and we take it from there. We do the codon optimization, gene synthesis, transfection, expression purification, and deliver the purified antibodies and the QC data to you as per requirement. And with the MamPower package, we also deliver the antibody heavy chain and light chain synthetic genes in a 57 vector, along with a detailed sequence, optimized gene sequence report. Do you accept all antibodies for your high throughput service? So our high throughput service is applicable to human IgG1, human IgG4, and mouse IgG1 kappa light chains only. Of course, if you have other requirements or different isotype, it does not mean that we will not consider your request. We'll certainly work with you, but it may not be eligible for the pricing of this high throughput service. So we might have to use our customized antibody production services if you have any special requirements or preferences. Excuse me. For your glutamine synthesis cell line, do you perform mycoplasma testing, and what method do you use? Yes, absolutely. We do perform mycoplasma testing. We use both PCR-based methods and Hooke's staining for mycoplasma detection. Excuse me. Excuse me. Do I really need antibody production, recombinant antibody production? Can I use my hybridoma for antibody production? That's a great question. You can certainly use hybridoma for antibody production. In fact, the production of antibodies was once limited to hybridomas, but actually they are unsuitable for large-scale production because the yields are relatively lower. I believe they can be less than 100 milligrams per liter, and the integrated viable cell count in bioreactors is also quite low, and so they are not really suitable for large-scale production. But I guess it all depends on your end application, its sensitivity, and the need for reproducibility and how much you need. And we do know that customers have used hybridoma for antibody production. For example, if they need it just for a grant application, then they just use hybridoma. So it all boils down to what your requirements are and what your ultimate application is. So what does your production team do to optimize antibody expression? This is something that we covered earlier. Basically, our scientists rely on their accumulated experience in recombinant antibody expression based on their knowledge of sequence and the relationship between sequence and function. And since sequences between individual clones could vary a lot, for example, sometimes they may have the wrong scaffold, production team checks to see if the sequences are normal. And sometimes they can also improve expression levels, without altering the antibody function, by manipulating the constant region and framework region, of course, in discussion with the clients. In terms of optimization, we can also try a bunch of other things. We can change the host cell line. We can change the signal peptides, change the expression vectors. Of course, codon optimization would also help. Sometimes we can optimize the transfection and purification protocol to achieve the desired results. Which type of purification methods are you using? So for recombinant antibodies, we use protein A resin. It's one-step protein A purification. And again, it depends upon your purity requirements. We can certainly do multiple purification runs depending upon how pure you need your sample to be. Why conduct developability assessment at this stage? So that's a great question. I'm not an expert in that subject matter, but I'll try to address that nevertheless. So as you know, developability is the likelihood of successful development of a lead candidate into a stable, manufacturable, safe drug. So why do it early? Why do it in a high throughput format? So if you can employ a set of small scale, fast and predictive tests, that can address the biochemical and biophysical features that would help you to identify a clinical candidate molecule at an early stage in your pipeline, that's really advantageous. And so, high throughput characterization will help you assess, for example, aggregation or stability of your antibody. And that will give you an early picture, that will help you identify an antibody with superior biophysical properties. So other than accelerating your overall workflow, I think it will also help minimize any downstream risk. So, it's good to incorporate developability as is early on in your workflow. So that's about it, I guess. And before we end this session, and there seem to be no other questions, I'll just go through this panel again, see if there are any questions that I was not able to address, and we'll make sure we get back to you. I also want to let you know that GenScript will be attending AACR and PEGS this year. So if any of you are going to any of those conferences, please stop by the GenScript booth. We have a large contingent of our sales staff, marketing, technical support, and even production team scientists who will be at these booths to answer any questions that you may have, and overall help you with your projects. So that's it for now. If you have any other questions, comments, or feedback, you can certainly email me. I provide my email ID in a slide earlier. And that's it from the GenScript team. On behalf of all of us, I wish you all success in your research. And once again, thank you for your time and attention.